In this video, we're going to look at how we can manipulate DNA for our own purposes in a research setting. We're going to talk about how to clone DNA in a host organism, DNA libraries, PCR to amplify DNA, and recombinant DNA technologies in general to express protein or to make transgenic organisms. So just to begin with, if we want to use recombinant DNA technology, which is also called molecular cloning, which is also called genetic engineering, we basically have 4 steps to do. And these 4 steps are, we need to get a pure sample of DNA, our target, and we need to amplify it. And we're going to talk how we do that using restriction enzymes and PCR. We need to incorporate that fragment into another DNA molecule. That other molecule is called a vector. And when we have the vector with our DNA, we need to put it into a cell to replicate it. And then once we have our vector inside the cell, we have to be able to distinguish those cells from cells that don't have vectors. So we're going to talk about these 4 steps on how to obtain and amplify a segment of DNA. So first, let's talk about a vector, or it's also called a plasmid. In general, plasmids can range anywhere from 1 to 200 kilobases, and it's circular DNA, usually found in bacteria or yeast. Now, these plasmids are what we're going to put our DNA of interest in. We're going to insert it into this plasmid. And in general, plasmids have features that are common among generally all plasmids. First, they have some kind of resistance to an antibiotic, and this will be useful in screening, which we'll see in a little bit. They also have segments that can turn on gene production. Here we have those segments called LAC-Z and LAC-I. I'm not going to go too much into depth on these right now, but basically you add a molecule in, and once that molecule is in, then your gene is going to be produced. That is, the gene we insert in this plasmid, the protein will start being made. These plasmids also have multiple restriction sites, because to insert our plasmid, we have to cut open this DNA. So the DNA must contain restriction enzyme sites. Now, plasmids in general have hundreds to thousands of copies in any given cell, which is about half of the cell's total DNA concentration. And generally, plasmid, you can only put a DNA sequence that's 10 kilobases in length. And here is a very common plasmid in molecular biology or biochem. It's called PUC18, and it usually goes into E. coli. Now, if you don't want to use a vector, you can also use a virus or a bacteriophage. More specific, bacteriophage lambda. And this is good because you can actually put more DNA into a bacteriophage. You saw in the vector we can do 10K, in the bacteriophage we can do 16K. Then once we have our DNA of interest into the virus, we let that virus infect bacteria, and then our bacteria will have our DNA of interest. And if we want, we can also use this to infect insect cells if we're trying to do research in insects. And the idea of how it works is, here's the phage DNA, and there's an attachment site between the phage DNA and E. coli DNA, and that will insert our gene of interest, this pink part, into E. coli, and now we have a transgenic organism. But let's say that our DNA insert is really big. So let's say our DNA insert is over 100 kilobases. Then we can actually put them into what are known as BACs or YACs, so bacteria artificial chromosome, or yeast artificial chromosome. So since yeast are eukaryotes, they have linear DNA, while the bacteria artificial chromosome are just more or less giant vectors, only 1 copy per cell, but they do allow us to get larger DNA inserts for molecular cloning. So how does this process work? So here's the diagram that we're going to follow. So here's our vector we want to get our DNA into, and the blue and green lines are our DNA of interest. So the first thing we need to do is we need to cut both the vector and our DNA. And usually you will use the same endonuclease on both the vector and the foreign DNA, because if you use the same nuclease, you will have the same overlaps, the same DNA overhangs, right? And you want to use sticky ends, so yeah, your DNA will go in the correct 5' to 3' direction. If you use blunt ends, your DNA could go in backwards, and in fact it will go in backwards 50% of the time. So you allow your vector and your DNA of interest to naturally come together, and then you use a molecule called ligase to seal up and combine the DNA. So what ligase is, it's an enzyme whose job is to connect DNA strands. That's all it does. And so when we're combining our DNA like this, we're saying we're annealing our DNA. And ligase, for it to work, it uses ATP. And it's the same idea with bacteria. We have our phage DNA. We cut out a part of that DNA that is not required to infect the bacteria, because we need it to infect bacteria, so we don't want to mess with that portion of the DNA. But other than that, it's the same. Restriction enzymes, ligase, put that into a virus, and you're ready to go to infect your bacteria. Okay. So let's say we have our vector with our DNA, and we put it into bacteria somehow, either through a virus or through a process called heat shock. So what heat shock is, you take your bacteria and your plasmid, and you combine them into one test tube, and you put them on a heat block that is roughly 42 degrees centigrade. And you do that for 30 seconds. So you mix your plasmid with your bacteria. And during these 30 seconds, since the bacteria are under stress, under high heat, what will actually happen is that the bacteria will open up their plasma membrane, and DNA will be able to go in. And we hope that DNA is our plasmid. So after the 30 seconds, you then put the bacteria on ice for 2 minutes, and by putting them in ice, you're allowing the bacteria to reseal the holes in their plasma membrane. And then you plate your bacteria, trying to get your bacteria with plasmid to grow. Now, really, doing this method or doing the virus method, maybe only 0.1% of your bacteria will successfully uptake your plasmid. That's okay, because as long as you have one cell, all it takes is one cell to grow your vector. Once they're on a plate of medium, or media rather, they will multiply and they will grow. But we want to make sure that we only have bacteria that have our vectors. And so to make sure that is the case, we use what are called selectable markers. The selectable is we're selecting for our vector. And there's 2 ways to do this. One, antibiotic resistance. So if you remember back to our slide on the puck vector, it had ampicillin resistance in the vector. That means that any bacteria that has our plasmid is now resistant to the antibacteria ampicillin. And so when we make our plates, which is basically bacteria food, we put the antibiotic ampicillin in our plate. And the idea is any bacteria that does not have ampicillin resistant dies, while any colonies that we see have our vectors in it, because they must be resistant to ampicillin. Another common way for a selectable marker is using beta-galactosidase, which is called blue-white screening. And the way this works is that on our vector, we have an enzyme called beta-galactosidase that is being made. And we can add a chemical called X-gal. Now, organisms that have beta-galactosidase will cut X-gal in two to make galactose and then this compound that is blue. Any organism that does not contain this enzyme will not turn blue. So the colonies here that are blue must have our vector, while the colonies that don't remain white. And so just some terminology for you. So each one of these spots is a colony, or you might hear it called a plaque. And in general, we call all of these colonies a lawn. Now, after you pick your colony, and you pick it up and you put it in media to grow the E. coli more, you must verify that your vector actually has your DNA sequence in it, because it is possible to get false positives. That is, it may be blue, but for some reason it doesn't have your vector. So you need to use DNA sequencing to make sure your vector has your protein or DNA sequence of interest before you carry on with any more experiments. All right. That's it for this video. I will see you in the next video.